previously we have in the first lecture, we have covered what is fermentation, right? So this course is basically about fermentation. So you have to always revise and remind yourself what is fermentation, because if you don't remember what is fermentation, then it would be problematic for you to follow or to understand the following topics, okay? So fermentation, basically just to give you a quick revision, fermentation is a process of growing the microorganisms, right? And you give them food, but in the laboratory we call media. Media is actually the food. Medium is the same thing. Both words just differ in terms of singular or plural. Medium is singular, media is plural. So they are used in different contexts, okay? But they refer the same thing, nutrients or food. Right, so you you give media or food to the microorganisms. You grow them by giving the food, and then you put them in a platform, right? Under certain conditions or environment. So what is meant by the condition is the pH, the temperature, pH, temperature, oxygen. Yeah? So those are the environment that you give for the microbes to grow well. Yes, and the reason why you do the fermentation or why you control such things is because you want the products of the microorganisms, which is the fermented products. Okay, and what kind of products? It depends on the microorganism. Different microbes will produce different products. Okay, for example, Saccharomyces cerevisiae is a very well-known yeast that can produce ethanol. So ethanol is the product. So at the end of the day, we want to have the products because we want to use the products for many purposes, like for ethanol can be used as biofuel. Ethanol can also be used as sanitizing agent, right? What else? It's not just ethanol. It can be many things like enzymes, food, even food from the fermentation, and then drugs, yeah, vaccines, and so on. So there are so many products that can be produced from fermentation. So that is the reason or the motivation why scientists carry out or do the fermentation. And in this course, we are focusing on artificial fermentation. So fermentation, you know that they can happen naturally or it can happen in the lab, the one that we control. The one that we control is when you grow the microbes, you take care of it, that is artificial. That is what you manipulate. But this natural fermentation, it happened without the human control. Like for example, in your gut, it's also called fermentation because there are microbes, there are food, and also there are platform. There are certain conditions, whether acidic or not. And then the products from the fermentation in your stomach is the simpler components. Is actually the digestion. The digestion is a fermentation process. And then apart from it, natural fermentation, when we say composting, you put like perhaps if let's say the food waste, if you put it, if you just dump it on the soil, and it will degrade. It will take some time, but it will degrade naturally. There are microbes in the soil that could degrade the food waste. So that is another example of natural fermentation. But in this course, we are focusing on fermentation that, sorry, I forgot to record the video, the lecture. Oh, it's good. Okay. What point did I mention this on? Oh, yeah, okay. Fermentation, natural fermentation. Okay, so the one that in the lab is the artificial fermentation. So in this course, we are focusing more on the artificial fermentation, the one that is in the lab. Okay, anything in the lab is basically artificial lah. So it's like artificial environment because you are providing the environment that is not natural actually, because the microbes come from outside, from the environment, right? So if let's say you don't isolate it, it will just like live in the soil, in water. But when we bring it into the lab and we try to grow it, so we are actually providing an artificial environment to the microbes in order for them to grow, and we want to harvest something from them. Okay, so that's why in the lab we call it artificial environment. Okay, it's clear, right? I just want to make sure that I didn't go into waste of the lectures. Yes, huh? Okay, fine.
Yes.
Thank you, thank you. So that is what we have studied in the first topic, fermentation, what it is, what are the important factors of fermentation. And then in the second topic, I expect you guys to read about media, yeah? What are the requirements for growing microorganisms in the lab, okay? And when we talk about media, you have to know what are the important requirements. Yeah? So microbes are living things. They need different nutrients like human, okay? Like they need carbon sources, nitrogen sources, sulfur, phosphorus, and other nutrients like trace elements. So the requirement, the amount of it, it depends on the type of the microbes. When we say microbes, it's a very broad category of organism. It can be bacteria, it can be yeast, it can be fungi. Okay, so from now onwards, you guys are in third year, right? So you have to be careful with the term that you use. For example, when we say bacteria, what is meant by bacteria? You must know, for example, like Escherichia coli, Bacillus subtilis. Is it a bacteria or is it yeast or is it fungi? So you should know that bacteria is what, okay? Yeasts are what? Pichia pastoris, Saccharomyces cerevisiae. Is that what kind of organism is that? They are yeast, okay? And then fungi, Aspergillus flavus, Aspergillus niger, Aspergillus teres. So those are examples of fungi. The names are scientific names. You have to differentiate what is what actually, yeah? And when I say microorganisms, it refers to all of them, bacteria, yeast, fungi, okay? Because fermentation can be run by different microorganisms. It can be bacterial fermentation, yeast fermentation, fungal fermentation. Okay. But now we are just talking about when we say fermentation in general, microbial fermentation. Any type of microbes, it covers every type of microbes. Okay. So in the second topic, you studied about the media, what are the requirements, how you prepare the media for growing the microbes. Of course, you cannot just keep it just like that. We have to sterilize it, because the reason why we sterilize the media before we give to the microbes is because we do not want the media to have other things, because we want now to just focus on we just want to grow our microbe of interest. We want to ferment our microbe of interest, right? For example, if you have Escherichia coli and you want to grow it, you want to ferment it because the bacteria has the gene of, for example, amylase. So at the end of the day, you want to harvest, you want to get amylase from the microorganism after you run the fermentation. So of course, you do not want any other microbes to grow together with your microbe of interest, bacteria of interest, which is E. coli in that case. Because we know that in the environment, there are so many other microbes, right? So that's why we have to sterilize the media. And also not just the media, but also the platform. So it depends on whether you are using shake flask. So if let's say you are fermenting in the shake flask, you have to sterilize the shake flask as well, together with the media. And only then you can put, the inoculum or the seed culture into the sterilized media and shake flask. Okay, so that is how you handle or prepare the media for the fermentation. Okay, so that is the second topic, the media and the preparation and the important elements, the requirements, the basic requirements and so on. So in this LU, in the third LU, we're going to talk about bioreactor. I mentioned about bioreactor in the first lecture. So bioreactor is also called fermenter. So both are refers to the same thing. It refers to a platform where fermentation occurs. Fermentation is carried out. Reactor is also, when we say only reactor, bioreactor or reactor can also be used, but for biotech purposes, we don't refer to reactor that really much. We use bioreactor or fermenter more, the terms, because if we say reactor can be a general reactor. It can be used for just chemical reaction. Yeah? So in biotech, we are concerned with biological reaction. Yeah, that's why we are in biotech. Yeah? Because we are dealing with biological things. So the reaction is biological reaction. So it's not just a normal reaction. Okay, so that's why if you see most of the terms like reactor, there is the word bio in front. So it refers specifically to the reactors that are used for carrying out the biological reactions. So if just reactor, if people in chemistry, they also have reactors, right? First, they need to do like large scale chemical reaction, for example. So the one that they use is just reactor. So that is the difference. Same thing goes to like bioethanol, ethanol. What is the difference between ethanol and bioethanol? So bioethanol is ethanol that is produced from biological reaction or from fermentation. That is the difference between just ethanol. Ethanol can be produced from chemical reaction. Do you know that? Yeah? So you have to know that the difference between all those things that have bio in front. Okay. So that is quite understood. You know, bioreactor. I mean, like for example, bioethanol. It should be produced from biological reaction. So that is what we are going to cover. Okay. The design and features of bioreactor. So what are the things that we need to know in this topic? The types of bioreactor, fermenter is the same thing. Yeah? If I can just use later fermenter, bioreactor, it refers to the same thing. So there are many types of bioreactors. So here we are going to discuss stirred tank bioreactors, airlift bioreactors, loop reactor, disposable bioreactors system, and immobilized bioreactor system. And the difference between each of these is actually the mechanism. So what is meant by mechanism is how the bioreactor operates, okay? Whether it's by stirring or whether it's by aeration. So we have a look at it later. And then we're going to have a look at what are the fundamentals of bioreactors. So when we say bioreactors, what are the things that we need to know or the important aspects of bioreactors? And then sterilization, okay? Sterilization of, this is one important step to prepare the bioreactors for the fermentation. And when we talk about bioreactors also, another important aspect is forming, okay? So forming is the formation of forms. So when such things happen like chemical reaction, biological reaction, right? Even in the shake flask, there is foaming. So we talk about it at the end of the topics. Okay. So let's have a look at the introduction, some introduction of bioreactor. Okay, so this is just to recap what is fermentation. So please remember very carefully what is fermentation, because this is actually the core process that all the topics after LU1 is all about. Okay? If anything unclear throughout this lecture, please just shout out, because I might not realize your comments in the chat box. Please just speak up, just chip in, so that I know that I don't talk alone. Okay. Okay, thank you. Everything is clear. Okay, fine. So just a recap for what is fermentation. When we talk about fermentation, there must be microorganisms. Without microorganisms, it's not called fermentation. Okay, because the process is about growing the microorganisms, right? So microorganisms can be bacteria, yeast, fungi. Yeah? Right. So another thing is media or media, media, food. So this is the food lah. That's the reason how the microbes can grow. Without food, there will be no fermentation as well. Okay. And then another aspect is platform and condition. So platform is refers to bioreactors, the place where we carry out the fermentation. Conditions is the environment, like the pH, the temperature. Because if you say about platform, this process is a closed process. So it can't be open process because otherwise it will be contaminated, from the things outside. So it should be a closed process. And of course, when it is closed, it must have certain conditions, right? It is closed. So what is meant by the conditions here are the temperature, the oxygen level, if it is aerobic fermentation, pH, the nutrients, the media, and so on. Okay, so that is meant by conditions. Platform is referring to the bioreactors or fermenters. And the last one, of course, the product. So that is the motivation why you carry out fermentation, why you grow the microorganisms, because you want to harvest certain products, products from the microbes. So the product from the fermentation is called fermented products. Okay, so we are at the third point, the platform, which is the bioreactors. Okay, so bioreactor is a place where bacterial, fungal, animal, or plant cells are grown for their products. So apart from the microorganisms, if we talk about biotechnology, we are not just dealing with the microorganisms, but also animal and plant cells. So animal and plant cells, they are not called microorganisms because they are they are big in size. Micro is the one that you cannot see with your naked eye. Okay, you can only see with a microscope. So that's why it's called microorganisms. But but in biotech, we also concerned with animal and plant cells, apart from the microorganisms. So there are also bioreactors. Bioreactors, they are designed whether they are for the microorganisms or whether they are for animal and plant cells. Okay, so it depends on the type of organism that you are dealing with when we talk about bioreactors. Okay, so basically it is defined as a platform or a place where microorganisms, animal and plant cells are grown for their products. Okay, it is an equipment which provides the control environment in order to achieve the optimum growth. So the good thing about bioreactor is that we can control the temperature, we can control the pH, or we can control the oxygen if it is aerobic fermentation inside the platform. Okay? So the reason why we control it because microorganism or animal plant cells, they have their own optimal temperature, optimal oxygen level, optimal pH in order for them to grow happily, in order for them to grow optimally. Optimal is like at the best level, optimum growth. So the difference between optimal and not optimal, okay, if let's say you grow the microbes, let's say E. coli grow best at 30 to 37 degrees Celsius. If you grow them at 28 degrees, okay, let's say 25, which is below 30, which is the optimal growth rate, the optimal temperature for E. coli. At 25 degrees, E. coli still grow, but it doesn't grow that happily. It doesn't grow that really the best, okay? So perhaps if let's say that E. coli carries amylase gene, so at the end of the fermentation, you expect the E. coli to produce amylase. If let's say you have two conditions, one is at 37, which is the optimal temperature for E. coli. And another one is you grow E. coli at 25 degrees, which is not really an optimal temperature for the E. coli. So in both conditions, the E. coli will grow, but the difference will lie in terms of the products produced. Okay, maybe the one that is grown at 37, it will produce a more high amount of amylase. a high amount of the product compared to the one that is not grown at the optimal temperature. So that is the difference between just grow or optimal growth. Yeah. Okay. So if let's say the microbes are not, if the organisms are not at their optimal temperature, optimal pH, optimal oxygen, they still grow, but maybe not really happily. Okay, so at the end, they don't produce that really much products. So that's why it's very important to achieve the optimal conditions for the fermentation for every type of organisms that you grow in order to get the best product. Okay, so bioreactor, fermenter is the same, yeah. So when we talk about bioreactor, in the lab, it starts actually this fermentation. It starts with shake flask. Shake flask is also called fermenter, literally, but normally when we say fermenter, we always refer to this advanced machine or equipment, which is the real bioreactor lah. But by definition, shake flask, as I said just now, fermenter is a place where fermentation happens. So shake flask is also a platform of fermentation, but it's also called fermenter, but not that really commonly used. When we say fermenter, you have to know that it's something related to the machine, to the equipment, to the advanced one. But fermentation can also happen in shake flask. So, and even actually the studies, any fermentation studies, it starts with shake flask because that is the benchmark, that is the starting point. You cannot go directly to the bioreactor, although that, of course, bioreactor is the best platform. But when we want to develop any new fermentation studies, for example, you just like isolate new bacteria from the soil and you want to ferment it, okay, you have to start with a shake flask because that is the basic point for fermentation in the lab. Okay, so shake flask is a very well-known platform for conducting fermentation. Yeah, and before people invent bioreactor, advanced or all these sophisticated machines, bioreactors, people start with shake flask. Yeah? People start with shake flask, and from there they have an idea how to improve the platform. Okay, so and even until now, like when I was doing my PhD, I still start with shake flask fermentation as the starting point for the fermentation studies. Okay, so it's very important for you to know that shake flask is the crucial starting point for fermentation in the lab. Okay, so shake flask, right? This is shake flask, Allen-Meyer flask. It's a conical flask. Yeah? So what does it has? It doesn't have all the control system. Of course, you can put it on incubator, whereby you can control the temperature of the environment, but it doesn't have the pH control system. It doesn't have the oxygen control system. Okay, so that's why people invented bioreactor actually, because people want to have a better control system for the fermentation. Okay, so the limitation associated with shake flask, there is no pH dissolved oxygen control system. Okay, less efficient mixing, especially for large volume. Okay, for small volume is fine, not that really bad, but for large volume, if let's say, because these shake flasks, there are many sizes of shake flasks. There are small size, 250 ml, and then 500, 1 liter of shake flask. So if you are doing like 1 liter fermentation, if let's say you are using 1 liter flask and you are carrying out like 500 milliliter in that flask, so the mixing might not be that really efficient because the volume is already a lot. And the capacity for the incubator, this shaking incubator, this is what we call shaking incubator. You put the flask on the incubator and it will shake the things. Okay, the capacity for the shaking is limited because this incubator is actually designed for a small shake flask. Okay, I think the maximum is about 250 RPM. RPM is the speed. So the larger the speed, the the more intense the shaking. But for the incubator, is limited to 250, if not mistaken. Depends on the incubator. So it's quite limited. So it might not be giving the optimal mixing for big or large shake flask. Okay. Okay, and then limited scale of operation. Yeah, shake flask is normally used for preliminary studies and not for production scale. Okay, so if you're talking about scale, scale ni lah skala kan. So it has a small scale. So if we want to first study about new strain, new microbes, we have to start with a small scale, or we call it preliminary scale, preliminary studies. Okay, preliminary studies or scales use shake flask with about like less than 1 liter scale. So that is quite that is considered small scale. Okay, but when we want to produce, like after, let's say you have optimized everything, you know already what is the best condition for the fermentation, and perhaps you want to do large scale fermentation. So shake flask is no longer suitable. So that is when you have to move to bioreactor. Okay, right. So still, I want to emphasize on the limitations of shake flask in this point actually. Right. So due to the limitations of the shake flask, the scientists invented advanced platform for fermentation, which is called bioreactors. Yeah, because bioreactors. This is an example of a bioreactor. The model or the look of bioreactor, it can be different because there are many models. This is just one example. It's not the only type of bioreactor, but it's just one example of bioreactor. It's actually the same thing. Like if let's say you will go for cars, there are many types of cars, many brands. Same thing goes to bioreactors. There are many brands, many scales, and many models. So they are also designed based on type of the organisms for the microbial, or whether it's for animal or plant. So this is just one example of bioreactor. You can see that this is what we call vessel. Okay, so there are two units, two components. The first one here is the vessel where the fermentation takes place, and this vessel is connected to control unit. So you can see the control tower over here, and they are the advanced reactors now. They have the touch screen, and also you can actually connect it to mobile devices in order for you to observe or monitor your fermentation from your mobile devices. So that is the one that are advanced bioreactors. Okay, so the reason why it is connected to the control unit is actually for us to monitor, because on the touch screen here you can set whatever the set point of the temperature, for the pH, for the oxygen, for the mixing, and so on. Okay, so this is the vessel is where the fermentation takes place. So the traditional bioreactors, it just have the vessel. It doesn't have advanced control unit kind of things. Okay. So the difference between the shake flask and the bioreactor. So the main differences is in terms of the control conditions. Yeah? As I said just now, the shake flask doesn't have the pH, temperature control things. And not the temperature. Temperature can rely on the incubator. The pH, the oxygen, and also the mixing is just by shaking. Right? Whereas for the bioreactor, the mixing mechanism is by agitation. Okay, so there is a difference between shaking and agitation. So when you say shaking, so for example, if you have a bottle of water, right? A bottle of water, and you shake it, that is shake. Okay, so meaning to say that inside the bottle, there is nothing that can help. There is nothing like spoon or what, or something like stirrer. It's just shaking. It depends on the, the movement of the bottle, right? So that is how shake flask. That's why it's called shake flask. It's because the mixing is based on the shaking. Okay. So the mechanism of mixing in shake flask is by shaking, whereas bioreactor, you don't shake the bioreactor because there is a stirrer inside it. Okay, we are gonna have a look at it. Like how you imagine it, like if you have a mug of water, you have the spoon inside. the teaspoon, and that is the one that you use to stir, right? So stirrer, right? So that is like how agitation mechanism is. You have something that facilitate the mixing inside the vessel. So that is what happened in Bioreactor. Okay, so I'll go into detail for the mixing in Bioreactor after this. Okay. Performance of any bioreactor depends on many functions. Okay, it depends on the biomass concentration. So when I say biomass here is the cell concentration. Okay, the cell here refers to either is bacteria, yeast, fungi, or animal or plant cells. Okay, but in general we just say biomass. Biomass means the cell. So throughout the fermentation, the cell must be high. Why? Because of course you must have more cells in order to get more products, right? So that is one of the important thing that you have to take into consideration. Okay, so that determines the performance of fermentation in the bioreactor. And then sterile conditions, which must be maintained, right? So the whole system here is a closed system, right? And it must be sterile. So how to make sure it is sterile? Before you start the fermentation, when you prepare the media and you put and the media if let's say you are using bioreactor, it needs to be put in the bioreactor, and the whole bioreactor and the media. This is called vessel. The whole thing here, the glass vessel. The whole thing here should be sterilized. So one of the common ways to sterilize lab stuff, the equipments and the consumables of the media in the lab is by autoclaving. Okay, so sterilization is a general concept. When if you want to sterilize the top bench, right? You spray the alcohol, so you sterilize the top bench, right? But when we say autoclaving is one method. Is a method of sterilization. The whole process is sterilization. Autoclaving is just one kind of method. Okay, you have to differentiate it. Sterilize, when we say sterilize, it can be any methods. It's not just autoclaving. Okay, but in the case of media and equipments or consumables in the lab, normally we sterilize all those things by autoclaving it. You guys know what autoclave is, right? Autoclave. Do you know? Okay. Yes. So autoclaving is because the reason is because the method depends on the heating, the heating of the equipments and the things that you put into the autoclave. So that's how the sterilization happens. Okay, takes place. Okay. Performance also depends on the effective agitation, distribution of nutrient and microbes. So just now I talked about the mixing mechanism. Mixing is like how the things are mixed inside the shake flask or inside the bioreactor. So the reason why mixing is very important is because you do not want your culture, if you have, let's say, in the shake flask, just imagine shake flask, you don't put the things just static, because you want the nutrients and the cells to go like throughout the working volume, so that the cells get enough nutrients, right? But if let's say you just put the things statically without any mixing, so what happened? There would be sediments at the bottom of the culture, right? So what happened at the end, fermentation will not happen optimally because there is no mixing. So mixing is a very important aspect of fermentation, liquid fermentation. So having said that, the agitation, the mixing process must be effective so that the nutrients and the nutrients can be distributed uniformly to all the cells inside the vessel or inside the shake flask. Okay. And then heat control, removal and maintenance. Okay, and so when you carry out the fermentation, it is conducted at certain temperature. For example, at let's say 37 degrees. So 37 degrees is considered. The temperature is considered quite high. So it has all this, it generates heat throughout the process. So there must be I think the bioreactor, they have certain mechanisms how to provide the heat, how to remove the heat. It's not just about providing, but also how to remove the heat. Because otherwise, if let's say the heat is accumulated, so it would not be good for the machine and for the cells as well. So there must be a good way or mechanism of heat control, removal, and how to maintain the heat. Okay. Creation of the optimum shear conditions. Okay, so the reason why different bioreactors, they are designed for specific type of organisms. So some of the bioreactors, they are designed just for the microbial fermentation, and some of the reactors, they are designed for animal cells. Some are designed for plant cells. So the reason why we cannot use one type of bioreactor for all is because every organism, it has its own sensitivity of towards shear. Shear ni macam force lah. When you make something, there is a force inside the mixing system. Okay, so actually the agitation is not that really ideal. It's not actually suitable for plant cell. The agitated bioreactors, they are only suitable for microbial fermentations. And for microbial fermentation, it depends as well, because some of the microbes, they may have different shear sensitivity. Okay, so that is one of the important criteria of bioreactors. They must take into consideration the shear sensitivity of the organism that is going to be used inside the bioreactor. Okay, so high shear rates of bioreactors, it might be harmful to the organism. So like after this, we're going to talk about stirred tank reactor. It's not suitable for plant cells. Okay, low shear rates. Okay, so that kind of things are. Okay, the shear sensitivity is one important aspect that determines the performance of the bioreactor. Okay. So that's the intro about what bioreactor is and what are the brief important aspects about bioreactor. Okay, next we're gonna have a look at the types of bioreactor. What are the types of bioreactor? So here we are going to discuss 5 types of bioreactors. Stirred tank bioreactors, STR, or STB actually. And also called stirred tank fermenter, STF. So that's why I said bioreactor and fermenter, both terms are used interchangeably. So sometimes people say bioreactor, sometimes people say fermenter. So sometimes people denote it as STB or STF. Okay, if you read the articles, the fermentation articles, you will see like different terms used by the researchers, but they are actually referring to the same thing: bioreactors, fermenters. And then airlift bioreactors, loop bioreactor, and then we're going to have a look at disposable bioreactor system and immobilized bioreactor system. Okay. So let's have a look at first stirred tank bioreactor, or STR or STP. Kadang-kadang STB, kadang-kadang STR. It's the same thing. Okay, so in the lab, you can see that this is what we have in our lab, biochemistry lab. This is quite an old model. It's not really advanced one. If you can see the control unit, it looks like very old. Yeah, I think this is about more than 10 years ago, the technology, 20 years ago maybe. Yeah. But basically what you have to know about stirred tank reactor, the reason why it is called stirred, because there is something that is used to stir the culture inside the vessel. So the whole thing here is called vessel. Yeah? So just now I mentioned there are 2 important components of bioreactor again. So the first one here is the vessel, where you carry out the fermentation. You can see the culture inside the vessel here. And this one is the control unit. This is where you control the conditions like the temperature, the speed of the stirrer, the pH, and so on. But this one you can see is very old. The new one has the touch screen things, and it can be connected to mobile devices. But unfortunately, we don't have advanced reactors in our lab. So this is the picture that get from the wrapper. Okay, so what is the important components of a stirred tank bioreactor or stirred tank fermenter? So this is the vessel part. So the reason why it is called stirred tank bioreactor is because it has agitator or stirrer. That's stirrer. So stirrer is the one that facilitate the mixing. So that's why this the whole vessel is not shaken. Unlike the shake flask where you shake it, right? So this one is agitated, the mixing mechanism. So this is the stirrer or agitator, we call it. This is the one that mix the thing. So you can set the volume, the speed of the agitation, right? For this reactor, the maximum speed is 1000 RPM. So you can see that if you use shake flask, the incubator shaker, I think the highest speed is just 250 RPM. So that's why it is not suitable. So shake flask is not suitable to carry out large fermentation. So because of the limitation of the speed on the shaker. Okay. Okay. So this is the components. The stirrer is the one here. So the stirrer is connected to motor. So motor is the black stuff that you see over here. Can you see on the picture? This is the motor. It is the one that controls the speed. So you have to put it, it is actually detachable. So during the autoclaving, we don't insert the motor. You just take it out and you just autoclave the vessel only. Okay. So this is the typical configuration of stirred tank reactor. You can see the stirrer just now connected to motor, and you can see the layer over here. This is what we call cooling jacket. Okay, so it has the place where water goes into this jacket. It's actually water jacket or cooling jacket. So the function of this cooling jacket, the function of the water coming in is to for the temperature control. So that's how the temperature can be controlled and the heat can be released, can be discarded from the vessel. Anything more? Okay, the height to the diameter ratio vary between 2 to 1 and 6 to 1. Okay, so what is meant by the height to diameter ratio? The height is the height of the vessel. Diameter is the diameter here. So there must be certain certain ratio, like it's not just sebarangan the design. There must be a ratio of 2 to 1. So you can see that the vessel is the height is more actually than the diameter. Okay, basically normally is it can be much longer as well. So it can vary, the ratio can vary from 2 to 1 and 6 to 1. It can be 3 to 1, 4 to 1, 5 to 1, or 6 to 1. Okay, so that is the range of the ratio. The stirrer or motor, the motor can be top or bottom driven. So what is meant by top or bottom driven? In this case, it's top driven. So you can see the motor is on the top. It can also be at the bottom. So there are bioreactors that have motor at the bottom. So that is bottom driven. Okay, and each of that has its own advantages or disadvantages. Why we have to follow the ratio? Because in order for the reactions to happen, because the thing is like the design, it must there is a certain rule of the thermodynamics and so on. Like this is a bit engineering things. But if let's say you imagine a reactor that is wide, the diameter is big and then the height is small, something that is very, like very wide. So it is not suitable, especially for the industry. If you're talking about industry, it is so wide, it's something hard for you to control like large area compared to if it is tall. Okay, so that's why there must be certain rule of thumb for designing the reactors. So this is more towards biotech engineering or biochemical engineering, which is actually my field. My background is not to say pure biotech. I'm more to applied biotech. I'm more to a little bit more to engineering things. Yeah, thank you for the question. Okay, so you have to know that a certain equipment, they must follow certain design or certain rule of thumb. So there is a reason behind that. As I mentioned just now, it's much harder to control bigger or spacious surface or diameter compared to if you have like large, tall reactors. Yeah? Okay, so this one I mentioned just now, agitator. So the whole thing here, this is each of these, it can be impeller or agitator is the same thing. So is they have different types of impellers, where different impellers would create different pattern of mixing. That one is also engineering, but it's enough for you to know about the existence of agitator in stirred tank reactor. Okay, so that's why it's called stirred tank, right? It's something stirred inside. You can chip in, you can ask me the questions if you are not clear throughout the lecture. Okay. So this is, okay, just now we talked about ni kan, the whole vessel kan. So actually, the vessel, this is the thing what we call a head plate. So you put inside here, the vessel is actually just vessel. It's just the glass vessel or the there are also aluminium, stainless steel vessel, okay? But the vessel is just the thing, a container actually. But what makes the whole system here, this is what we call head plate. So this is where all the impeller, agitator is situated, is located. Yeah? So this is the things, and this is, as I said just now, is connected to the motor, right? This is the motor. And this is the motor is the one that controls the mixing. Yeah? So what this head plate has, so this head plate, it has, if you can see, there are many inlets. Inlets is the one, ada closure, ada inlets, ada lubang, and you can see. And that is where the temperature probe is inserted. So this hole, you can see there is a holes over here. There are many types of hole actually. So there are for temperature probes, temperature probes. Temperature sensor is different. The temperature sensor is the one that detects the temperature inside the vessel, but temperature probe is the one that controls the temperature inside the culture. Okay. Level sensor. So the level sensor is especially for during the fermentation. If let's say the level of the culture exceed the certain level, so it will give a notification to the reactor, especially for the advanced one, it can actually notify the person, like from the mobile devices. Okay? So the reason why we have this level sensor is because we do not want the culture to flow out from the vessel, because otherwise if it flows out, then contamination might happen. Okay. Another thing on this head plate, baffle. Baffle is for facilitating the mixing. Yeah. So it actually creates like certain pattern of mixing so that the mixing is more enhanced. Yeah, baffles. It helps the role of agitator actually. And then so there is a sampling tube. So this one is a sampling tube. It's a long tube connected towards the bottom of the vessel. So it's actually for taking samples. So during the fermentation, we have to take the sample, like depending on the culture, like maybe between every few hours, we have to take the samples. And this sampling tube is where the things coming out from the bottom to the top. So actually we suck it. So that's the sampling tube. And this is the impeller just now. This is the Ampliform sensor. Okay. So there must be also a sensor for forming in order to avoid excessive forming. So if let's say the forming exceeds, later we talk about forming. So it will also lead towards overflow. So we do not want the culture to overflow from the vessel. Okay, so that is the Ampliform sensor. So that is what head plate has. Okay, this head tape is actually the things that we put on the glass vessel that has all these elements of inlets and so on. Okay, so that is third tank reactor, the basic components of third tank reactor. So the second type of reactor is called airlift reactor. So airlift reactor. So what is the difference between airlift reactor and third tank reactor? So as the name implies, it depends on the air. It doesn't have stirrer inside this. You don't have any, like just now we talked about the impeller, the agitator, right? So this one is, it doesn't have the stirrer. So it depends on the aeration, aeration for the mixing purposes. Okay, so what is this airlift? It uses the action of aeration bubbles to create flow and as a means of mixing. Still mixing is needed because we still want the culture to be mixed properly, but the way how the mixing takes place is by using the aeration. Aeration is you provide the air, and this is obviously for aerobic cultures, aerobic fermentation. Okay, so this is examples of airlift reactor. So you can see this is Spudger at the bottom of the reactor. So the spudger is actually like is actually a ring containing small holes along the circle here. Kita tak nampak sini, but it has holes actually where the air that we provide to the reactor, that is where the air will come out through the culture here. Okay, so that's how the mixing can happen. Okay, it depends on the flow of the air inside the reactor. Okay, rising actions of the bubbles causes a circulation up and down. So this airlift reactor, this type of reactor is suitable for plant or animal cells that are very sensitive to shear. Okay, so plant and animal cells that are very sensitive to shear compared to microorganisms. Microorganisms, you can use third tank reactor because they can sustain the shear. So that's why third tank reactor is basically designed for microbial fermentation. But for plant and animal cells, we can't really use, especially for plant, we cannot use third tank reactor. So we have to use this type of reactor, airlift reactor, right? Because this one doesn't have stirrer, so it depends on the aeration. So that will provide less shear to the cells. Okay, the three types of airlift reactor, ALR basic loop, ALR with internal loop. So the second one here, okay, you can see here, basic loop is the one in the first picture over here. Internal loop is when you have something like this, right? The loop is inside. And then external loop is when the vessel has an external tube to enhance the flow of the medium. So this is external loop and on. Yeah? Or you can see also for this picture, this is internal loop. So you have a loop, internal tube inside the reactor. So you can see that the flow of the air is controlled like from upward and downward. You can see the flow, the arrow here refers to the flow of the air, right? Compared to if let's say it doesn't have the loop, it will be like the first picture over here. That is the basic loop. It doesn't have, the flow is random, okay? External loop is when you have external tube like this, yeah, to enhance the flow of the medium. Okay, so that is airlift reactor. Advantages of airlift bioreactors. Just now, it provides low shear, so it is suitable for those cells that are sensitive to shear, like plant and animal cells. So this is an example of algae cultures, algal cultures. Yeah? So this is example of algae being considered as plant because it's quite sensitive to shear. So we use airlift bioreactors normally to grow algae. Yeah? Okay, absence of agitated shaft. Okay, so just now we talked about stirred tank reactor. So the stirred tank, it has a stirrer. So one of the disadvantage or the challenge lah. I say the challenge with the stirred tank reactor is in terms of the sterility. So when you use the STR, the stirred tank reactor for many times, you have to make sure that before the fermentation it is sterile, it is clean, the stirrer. So you have to clean like thoroughly the things. But with the airlift bioreactor, because there is no stirrer, so that's one of the advantages, because we don't have to care about the things, the additional things inside the reactor. So it doesn't have the stirrer, so we don't have to make sure that like unlike if we use the STR, we have to make sure that the stirrer is clean every single time before the fermentation. Okay, that's one of the advantage of airlift bioreactor. Okay, extremely large size vessels can be constructed because the structure is actually quite much simpler than the stirred tank reactor. Yeah? So the construction of the reactor is much simpler. There is a question just on what type of ALR is used for algae, was it? It depends on what kind of situation. Normally is the basic one is already like suitable, is already sufficient. But if let's say you want to improve, you want to have like better kind of circulation, so you can consider the one with the external loop. Yeah? Right. So disadvantage, high capital cost to build large scale vessels, high energy cost to supply high air pressure, because it depends on the air very much. So the air, it means the oxygen, then, because we want to provide the oxygen. So oxygen might be a cost for it, because we don't provide the oxygen just for the growth, but also for facilitating the mixing. So it's actually more. Huh? Can we also use plant and animal cells inside stirred tank reactor? I mentioned this, not really, especially for plant. Stirred tank is not for plant cells, because the plant and animal cells, they are sensitive to shear. Shear ni macam force lah. So when you stir something, there is a force inside it, right? So the plant and animal cells, they are sensitive to that force. So what is meant by sensitive is that when they are sensitive, they are broken. The cells can be broken. So when the cells are broken, so the cells are dead. The animal and plant cells are dead. So that's why it's very important to take into consideration the sensitivity of the shear. So to answer the question just now, the stirred tank reactor is not suitable for plant cells. But for animal cells, they are type of impeller. Just now I mentioned about impeller, kan? There are many types of impellers actually, and they are certain type of impeller yang provide mild agitation. So some of the animal cells can be grown using stirred tank reactor, but depending on the impeller, like it's not the impeller, the one that is used for the microbial fermentation. Thank you for your question. That's very good. Okay. Okay, disadvantages of airlift reactors, high energy cost to supply. Okay, I mentioned this about the cost for the air, any oxygen kan tadi kan. Hard to maintain consistent levels of nutrients and oxygen in large vessels. Okay, that's one of the disadvantages as well. Formation of foam, okay, because of the flow of the air. When we provide the air inside the culture, inside the liquid, so it can create foaming. Excessive foaming, actually excessive foaming. So that's one of the challenges using airlift bioreactor. Okay. So this is algae tadi, as I mentioned. Okay, the third type of reactor is a loop reactor. So this one is a modification of airlift reactor. Just now we talk about first tadi ada reactor, ada apa ni, stirrer, right? And the airlift reactor, it doesn't have stirrer. It depends on the air for the mixing. For the loop reactor, it has the ALR element, but at the same time it has a small turbine at the base. Turbine is for the mixing, for the agitating, for the mixing purposes. Sorry. Okay. So basically, loop reactor is a modification of the ALR with a small turbine at the base, right? So you can see this is example of typical configuration of a loop reactor. So this is turbine, okay? And the air is still provided into it, like it's still like have the ALR element, but now with the turbine. Okay. So the turbine increases the outflow of the medium by sucking the medium down. So it actually facilitates the mixing, the turbine. Right. So the mixing is by the combination of the air and also the turbine. So that is loop reactor. It's not that really common loop reactor. The most common one is stirred tank reactor for microbial fermentation, and airlift reactor for normally for plant cells. So loop reactor is not that really common. In the lab, we don't have loop reactor. We just have stirred tank reactor. Okay, the fourth system is what we call disposable or single-use bioreactor system. Okay, it is a concept, the other concept of disposable or single-use. So disposable is something that you just throw. Single-use is like you use just for one time. It actually refers to the same thing. So this is actually a new concept introduced, like I think about more than about 10 years ago, 15 years ago. Okay. So what is this technology is all about? The reason why people introduce this is actually to facilitate the optimization studies, especially in the factory. Okay, so just now when we say stirred tank reactor, we can autoclave it. It is autoclavable. It is not single use. It is reusable. So you can use the vessel for many times as long as you sterilize it, right? But the problem with autoclavable, all these autoclavable STR is that it might take some time, the sterilization process, okay? And also the preparation of cleaning it, because when we start the fermentation, you don't just prepare the media, you have to set up the fermenter as well. You have to clean the reactor before this I'm talking about before the fermentation. You have to clean, make sure that the stirrer doesn't have the residues from the previous fermentation and everything is clean. And then you put the media in and then you autoclave. And that preparation, it takes some time, okay? And okay, that is the preparation. Now, when you start the fermentation, you do the fermentation, right? And after the fermentation, what happened? You have to sterilize back the fermenter. And that is what the process lah. You have to sterilize it. You have sterilize after the fermentation is to kill the culture, to kill anything that you want to discard after you harvest the products, because we do not want to endanger the microbes then after the fermentation. So that's why we sterilize again, we autoclave again, but the purpose is to kill the things, all the microbes. So you can see that there are preparation before the fermentation and after the fermentation. So those steps, depending on the type of the reactor, the bigger the reactor, the longer the time for preparing and for sterilizing it at the end. So I have an experience working with 2-liter reactor might take a few hours to autoclave, but if let's say you are using like 7-liter reactor, it might take half a day to autoclave for the preparation and for the after the fermentation. So it might take like 1 day to sterilize only, not the fermentation. So that is considered an obstacle, especially for industry. So that's the thing that people try to solve the problem by having this disposable system and single-use bioreactor. So disposable system and single-use bioreactor, you can see that basically there are actually plastic vessels that have been invented, that have been sterilized. So whenever the operator, like when, let's say I want to run the fermentation, I just use the bioreactor without sterilizing it. It's already sterilized. So I just use it directly for fermentation. And after I complete the fermentation, I throw it away. Okay, there is an issue about the waste and the plastic waste. That is something that is raised another concern lah. But just to introduce you the concept here, why people invented this disposable is actually to solve the problem of long operation of sterilizing just now. Okay, and they are concerned with the time. Why they are more concerned with the time? Because when we are talking about industry, right, the time is cost. So we don't want to spend too much time on the too much time on waiting, on preparing the things, or the all this lag time, because that will affect the productivity, affect the profit, and so on. So that is the idea, that actually the motivation of why people invent this disposable system. Okay, but just now, as I said, the issue of the waste, that is another thing lah, that might be another concern. But in the first place, that is the thing that you have to know, and that is what happened in the fermentation industry, the modern fermentation industry. People turn into this disposable or single-use bioreactor. So okay, I don't go word by word. I try to just emphasize on the main points. So the advantages of the single-use bioreactor system, there is no need to autoclave or sterilize or to clean the equipment before and after. Okay, right? And then minimum cost for startup analysis. No need to purchase full-scale lab fermenter. What's that? Just minimum cost for startup. Minimum cost for startup analysis. Ignore this point, I'm not really. Okay, reduce cross-contamination between batches. Okay, this is another important thing. Okay, for example, if let's say we use stirred tank reactor yang bukan single use, like the one that we use for many times. Although you have killed it, let's say after every fermentation you have autoclave it, not kill. You have autoclave it, you have sterilized it in order to kill all the cultures, right? But still, if you don't, let's say if you don't clean it properly, there might be some residues left, especially on the stirrer, on the impeller. So that's why the cleaning might take some time and energy. So if let's say the stirrer or some parts of the bioreactor is not cleaned properly, they might cause contamination. If let's say you use it for the next time and it's still there, the residues, so it will contaminate your culture. So that is meant by cross-contamination between batches. Okay. But if let's say you use single-use bioreactor system or disposable, because the things are new, for example, the plastic vessels are new, so you don't have such issues. There's no cross-contamination things, and people at the industry, they are very concerned because they are dealing with expensive products, so they don't want to risk, for example, like their process with using like reusable fermenters that can pose some risk of contamination. So they dare to use like the new one, even though they have to invest on the cost of the single-use bioreactors. But what matters more is the price of the products, or the media. Their media is expensive, so that's why they go for the single-use bioreactors. Okay. And then the advantage and minimum preparation of media. Okay, some of the media also, they come with the pre-sterilized one. It's not just the vessels, but also media. So that's meant by here. And then the single-use bioreactor system, it can be used for batch and continuous. So it actually behaves the same like the normal fermenters. So that's one of the advantages of it. It doesn't have limitation like it has the same performance as the normal reactors. Okay, so that's the single-use bioreactor. So this one is micro 24 reactor. So single-use bioreactor, it has many forms, many brands, many models as well. So this one is just one example of it, the one that I use during my PhD. Okay, and then the last step of bioreactor is immobilized system. This one is normally for animal cells, okay, because we know that the animal cells, they are very sensitive to shear. So the cells are taken care, like in this kind of reactor, whereby they are immobilized. So you can see, like, this is the reactor. It has the vessel still, but it has this sort of solid surface or gel that entrap the cells. So you can see the arrow here. The arrow is referring to the flow of the media. So it's actually the media flows into these immobilized cells. Okay, so that is immobilized system. And apart from the animal cells, it is also used for enzymatic reaction, whereby the enzymes can be immobilized onto the gel. Okay, the bioreactor it's not just for fermentation. When we have the enzymatic reaction, you can also use bioreactor. So, but the process is not fermentation because enzyme is actually not living things directly. It's derived from the cells. So it's not living things. So just to show to you that actually bioreactors can also use for enzymatic reaction apart from fermentation. Okay, so this is immobilized system mainly for plant and animal or mammalian cells. Okay, so those are the 5 types of bioreactors. Third tank bioreactor, airlift bioreactor, loop reactor, disposable bioreactor system, and immobilized bioreactor system. Okay, so make sure you know, especially the first 3 over here, what's the mechanism. Okay, the fundamental of bioreactors, what are the things that we have to take into consideration when we talk about bioreactors? I have updated the lecture notes, so later I will upload the latest, not the latest, the updated one, on Ellipse. So the one that perhaps some of you might download it from Ellipse. That one is not the revised one. I updated on the fundamentals part. Okay, so later I will update. I will upload the updated one on Ellipse. Okay. So when we talk about the fundamentals of bioreactors, what are the important things? So the design of the fermenter, the geometry. So just now I talked a little bit about the design, the ratio of the height to the diameter, right? Okay, so the design is very important, the geometry, right? So apart from the ratio just now, so the shape of the vessel, this is the vessel, right? So you can see actually the vessel is not, how to say that? It's not flat. Yeah? There is a curvature at the bottom over here. So there is a reason why there is a curvature or it's curved. It's not a flat bottom. Do you have any idea why? What is the relevance of having a curve at the bottom? Pressure, pressure not really. Flow, yeah, something related to surface area. Stability, yes, a little airflow, okay. Apa logiknya kalau let's say you have something yang bekas, let's say macam mana? Cawan, let's say cawan. Why it is curved at the bottom? Why it is not just like it has a hard sharp edge? Easy to remove. I see it's on. Apa tadi? Easy to remove. Just I tengok balik comment. Preserve heat. Apa lagi? Okay, thank you for the answers. Thank you for interacting with me. So some of you answered just now, easy to remove, prevent from scratch. Scratch, yes. Tapi the main reason is, okay, yang tadi kan cawan. Kenapa cawan dia ada bulat kat bawah? Why don't you have like a very sharp edge that tepi ni? You know, whatever the cawan or gelas. Yeah, easy to stir. Yes, that's one of the reason. I can accept that. But the most important thing is why? What do you think? You're dealing with culture. Culture ni ada banyak solids inside, right? It's not like a normal liquid. So you must make sure that the flow is good inside and also it doesn't accumulate, stuck at the edge tu kan. So if let's say you have a sharp edge, you know the sharp edge yang macam tak ada curve ni, so it will be easily to be stuck at the edge of the vessel too. That's why they create something like a curve at the bottom. So in order to avoid any stuck of the cultures. Okay, so thank you for the answers. That is the logic of the design, the geometry. So it's very important to have that sort of curved bottom. Okay, right. It improves mixing as well for stirring. Yeah, some of you answer for just now for stirring or for the flow. Yeah, it's something related to the mixing inside the vessel. Okay, it facilitates removal of the broth, especially when during the cleaning after, let's say, you have finished. So if let's say, just imagine if it has the hard edge, the sharp edge, so it's very difficult to remove at the corner over there. But if it is curved, concave design like this, it's very easy to remove. So that is the logic behind that. So that is in terms of the geometry. And then in terms of the what are the terms that we need to know. So I think in the practical question, I asked you what is defined working volume, isn't it? In one of the question. So what is the difference between working volume and total volume? Okay, let's say this volume, this vessel, let's say the total volume is 2 liter, it means that the vessel, actually by right, it can accommodate 2 liter of amount of liquid. But in fermentation, we don't carry out according to the total volume of the reactor. Okay, even though it has 2 liter volume, total volume, but we always have to reduce the volume of the working cultures. So that's how the working volume comes. The term of the working volume. Working volume is the one that refers to the amount of culture that we put into it. The rule of thumb, the ideal rule of thumb is 80%. So if let's say working volume should be 80% of the total volume. Okay, that is working volume. The volume of the culture that you work on, the liquid that you put it. Let's say you have 1 liter, so it should be just like 800 milliliter, which is 80%. Okay, and the rest of it is what we call headspace. Jadi we have head plate kan? Apa ni? Sorry. Mana yang? Okay, this one is called headspace. The space on the head, actually, the headspace. The headspace should be 20%. So the reason why this headspace is important, if you imagine your culture is inside here, this is headspace. So you cannot put it directly like the whole thing, because otherwise it will touch the top plate over here. Okay, and the function of the headspace is to prevent contamination. Okay, so whenever, if let's say your culture is very close to your top plate, this one is top plate kan tadi. So when it is very close, it means that there is no space and it will be easily flow out. Okay, and when it flow out, it will cause contamination. It will attract things from outside to come in. So that is the function of headspace, that is to prevent contamination by providing a gap. Okay, so that's why the rule of thumb is 20% headspace and 80% working volume. And it allows the foam air bubbles arising from within the broth to coalesce or break. Okay, so whenever the things are mixed, there will be foam on the top of the cultures. So whenever the foam or air bubbles are formed on the top of the surface, there is a space. Yeah. Otherwise, if let's say if you have the culture to the same level of the top plate, so there is no space for the foam or for the air bubbles, right? And then it will also cause contamination. Okay, and the third reason is to allow back pressure of air to build up and to improve the aeration of the broth. Okay, so if let's say the things are too compact, if let's say if you fill in the vessel with 100% of your culture, so it means that the pressure inside the vessel is quite high. Okay, so having the headspace, it allows the reduction of the pressure inside the vessel. Okay, and also it improves the aeration of the broth because there is a space inside it. So ada ruang lah. Okay, that is the geometry just now, the body of the vessel, and then the materials for the vessel construction. So the one that I showed just now on the picture, the vessel is made from glass. Yeah? So the reason why it is, and there is a certain type of glass, which is borosilicate glass. It's not like any type of glass. There is a specific type of it because the reason why, because we autoclave this glass, this vessel, right? So it must sustain the pressure, it must sustain high pressure and high temperature. So that's why there must be a certain type of glass, and the type of glass is borosilicate. The good thing about using glass, there is also a stainless steel. Yeah, let's see the glass first. So the good thing about using a glass vessel, it permits easy visual observation. You can see from outside what happened in your vessel. Yeah? That's the good thing about having glass vessels. And normally the glass vessel is used for small fermenter vessel or lab scale vessels. For large scale vessels, the material used is normally stainless steel. So the reason why stainless steel is because compared to glass, the stainless steel is more robust, right? And the construction is perhaps much easier compared to the glass if you are talking about big reactors, big fermenters. So it is ideal for large fermenter vessel. And the type is the type of steel is stainless steel, which is it doesn't corrode. Because of course the vessel, you put acid, you put bases inside the vessel in order to control the pH. So the steel must able to resist all kind of the harsh chemicals. So that's why the type of steel is stainless steel. It's not just any steel. Okay. Okay. But the problem or the limitation with the stainless steel is that perhaps it's not that really easy for you to see the content inside the vessel, unlike the glass vessel. So that's the limitation of the stainless steel vessel. Yeah. And then another type or another things that we have to take into consideration of bioreactors is the agitation. This is particularly for stirred tank reactor because only stirred tank reactor have the agitator. So this is how the agitator in the reactor looks like. This is a stainless steel reactor. Okay, so it's basically a long shaft consisting of impellers. So each of these is called impeller. Each of this component, it looks like macam fan punya tu kan. But this is what we call stirrer or impeller. Okay, so it's the one that drives the mixing inside. So the shaft is just for the impeller to be mounted. Okay, so those are the things. So there are many types of impellers. This you can see like this one is different. This one is with 3 blades. This one with 4. So there are many types of impellers, and it depends on the type of microorganisms that we want to grow inside the fermenter, because different impeller will give different shear level, shear force. So they akan bagi different ni lah, different sort of mixing pattern. Okay, so the one like animal cells, some of the animal cells can be grown using stirred tank reactor, but the impeller for animal cells must be not that really robust or not that really harsh as the microbe ones. It must be gentle, suitable for the animal cells. So what is the purpose of the agitation? Agitator is to provide mixing, of course, and mixing is for the nutrients to be distributed thoroughly, to all the cells inside the reactor, and also for the dispersion of air in the form of bubbles in order to supply oxygen for aerobic fermentation. So the mixing inside the vessel also facilitates the distribution of oxygen. Whenever you provide the oxygen, then with the mixing, it will be distributed properly or better throughout the cultures. And then just now, a question of constant living environment, pH, temperature, substrate concentration, this is the media. And then improvement of heat transfer between the medium and reactor. So the mixing system also facilitates the heat transfer between inside the medium and also between the medium and the reactor. So that's the purpose of agitation. So the drive configuration, it can be at the bottom or it can be at the top. Okay, so the one that I showed just now for the lab scale fermenter, it is top driven, yeah, the drive configuration. The bottom driven is more common for large scales. We don't have it in the lab, but it's just for you to know that there are two types of drive configuration. Okay, the rest of it you can just read by yourself. I just want to cut the time. Just want to highlight the important points, just to guide you throughout the learning unit. Agitator types. Yeah, so it's important to ensure proper mixing, right? Okay, so apart from the types of the impellers, what is also important about the impeller mounting on the shaft is the distance between one impeller to another. Okay, so this one has 3 impellers. Each of these is called impeller. So the distance between the impeller, there must be certain distance. I don't mention this one. The impeller diameter and distance, it should be 1/3 of the tank's diameter. That is the the impeller diameter. Sorry, the distance is, there is a rule of thumb for it as well, but I don't have it here. Okay, this one is 1/3 of the tank. This is different. So this is the impeller diameter, the diameter of the impeller. So how big the impeller is, it should be 1/3 of the tank's diameter. So the tank's diameter is the whole diameter here. So this one you can see is 1/3. Okay, and also the distance. I don't have the info, and I forgot what is the rule of thumb for it. But there must be a certain distance from one impeller to another. So you cannot put like too close, like, you know. So if it is too close, it will not cover the whole culture. Especially if, let's say, the culture is the size, the volume is big like this. So it should be, the reason why there must be certain distance is to ensure the mixing happen at all the points throughout the culture. And certain different types of impeller, it will provide different flow of the medium, different pattern. Okay, so that will determine a different level of the mixing, different quality of the mixing. So it really depends on the type of impeller, distance of the impellers, and also the diameter of the impeller. So those are the things that important in, when it comes to the agitator types. So these are the different types of impellers. Okay, so the most common one is 6-bladed Rushton, which is this one. Okay, so you can see there are many types. For ribbon black is, I think, is for animal culture. Yeah? So each of these, it has different pattern. It gives different shear or force. Yeah? So some of them give a very intense shear, or some of them are quite mild. So that depends on what type of organism that you are growing. Okay, that's enough about impellers. Okay, another fundamental type of another fundamental point about bioreactors is aeration. What is aeration? Aeration is the provision of air. This is for aerobic fermentation. Provision of air to the culture, to the fermentation. Okay, so how the air is provided is by this equipment or component called a spudger. Okay, so this is the a spudger. This is how it looks like. So it is like inserted inside the vessel, and on this a spudger, this one, you can see there are fine holes where the oxygen or the air comes out. Okay, so there must be a connection of oxygen or air that is provided to the culture. So the air comes out from that fine holes. Okay, so that's how the things or the bubbles can be created inside the culture. Okay, so aeration is normally we provide just either air. Air has oxygen, but oxygen is not in the pure form. But if, let's say, the culture is highly aerobic, means that the culture needs a lot of oxygen. So in that case, we provide pure oxygen gas to the culture. If it is just like not that really highly aerobic, then we just provide oxygen. Okay, so in some cases, other gases are also also provided using this Spudger. For example, nitrogen. Okay, in what case is nitrogen? Nitrogen is provided to the cultures for anaerobic cultures. Okay, anaerobic, the one that doesn't need oxygen, right? So in order for us to make sure that the culture, the vessel, doesn't have any oxygen, nitrogen is a sponge because nitrogen can eliminate the oxygen. Okay, so in that case, the nitrogen is sponge. Okay? Carbon dioxide is used for algae. For certain bioreactor, it has spudger, right? And also you can spudge the carbon dioxide for algae cultures. Yeah? Okay, that's it. So that's how it looks like, a spudger. Okay, so this is concerning the mass transfer of oxygen. So basically, this is like the process how oxygen is transferred from the gas bubble to the microbial cells. Okay, I just go very briefly here. Right, so the gas bubble that you see just now, like this one, the gas bubble ni. So don't forget that inside the culture, there are cells. You're growing cells, right? So that's why there are cells. So how the oxygen from the gas bubble is transferred to the cell, these are all the steps. So first it goes from the gas phase in the bubble, and then it goes into the gas liquid interface. So this is the medium, of course, the liquid. So dia akan go through this layer lah, actually, the gas liquid interface. And then it move into the 3-1 is the liquid film around the bubble. Okay. And then the fourth step is in the liquid, the oxygen, right? And then the fifth step is when it approaches the microbial cells. So there is another film, right, a layer between the liquid and the microbial cell. And then the sixth step is the cell liquid interface, okay? And the 7-1 is when it is inside the microbial cell, the oxygen transfer, intracellular. So that are the steps of oxygen before it reach the cells from the gas bubbles. And you can read by yourself more later. This is just stoichiometry. I think I asked the stoichiometry question in the practical, right? So perhaps you need to revise your chemistry about the moles, about this thing. Okay, I'll just skip this slide. Solubility of oxygen. Okay, so when we talk about solubility of oxygen, it's not to say that when you provide the air, the bubbles tadi kan, it will be transferred entirely. Actually, it's not. Actually, it's not. So there are things that actually affect the solubility of oxygen, yeah, before the oxygen can be consumed by the cells. Okay. So what happened actually, oxygen is 6,000 times less soluble in water than glucose. Actually, it's not that really easy. Just if you see the steps just now, it comes from the bubbles and then go to the liquid, and then it goes to all the layers, the interface, the film semua. That are all actually the obstacles that makes oxygen not that really easy to be dissolved inside the cells, or I mean inside the liquid, and to be taken by the cells. Okay. I think you boleh baca sendiri ke tak ni? What are the things that I need to explain here? Yeah, the important point is that's why when we talk about fermentation, we are talking about dissolved oxygen. Oxygen yang dissolved dalam liquid, okay, dalam culture. Not actually the total amount of oxygen because of the obstacles that I mentioned just now. So the oxygen is not that really easy to be dissolved actually. So contohnya you provide 100% of oxygen, it's not 100% that the oxygen will be used by the cells. it depends on how many amount of the oxygen that can dissolve. Okay, so that is why the term dissolved oxygen arise. Dissolved oxygen is the one that dissolved. Okay, this is some of the points relating to the oxygen supply. To ensure the balance between oxygen demand and supply, you must make sure that the oxygen uptake rate does not exceed the oxygen transfer rate. So meaning to say that the oxygen transfer rate must be more than the oxygen uptake rate. Uptake rate is the one that is by the cells, okay? OUR. How much the cells need, we must actually provide more than that. That's what the point here is. So what influenced the oxygen supply? Of course, the aeration, the spudger. This is the aeration, the spudger just now, right? The size of the spudger and the gas bubbles. So the holes here is very important, especially when we start the fermentation and we set up the fermenter. It's very important to make sure that the holes over here is not blocked. Okay, so those are the things that we need to do before the fermentation. Another thing that influences the oxygen supply is, of course, the mixing system. Yeah, the impeller, the size, the speed. Okay, if let's say the speed is not enough, perhaps the oxygen is not distributed properly throughout the cultures. So the mixing is very important. The velocity of flow, the air flow rate. Yeah? So when we supply the air to the culture, there is a certain speed of air. So they must be at certain optimal speed. If the speed is too low, then it might not be enough to provide enough oxygen to the culture. Okay, so that's the thing related here. Okay, this is just some of the example of works. If you see the research done in order to enhance the oxygen transfer in fermentation, it is actually a topic that people continuously try to improve in aerobic fermentation, how to improve the oxygen transfer. So why is it important? Because the oxygen is needed by the microbes for the aerobic fermentation, and it eventually it affects the product formation. So that's why it's a very important topic, oxygen transfer. Same as like media optimization. In fermentation, there are many important topics that people, the researchers try to have a look and try to improve from time to time. At the end, it's actually the same, to improve the fermentation, to get more products, more cells, to produce more products. Yeah? Right. Another important aspects of bioreactors is the heat exchange. Okay, I think I talked about the water jacket just now. So if you see this one, this is the vessel, and actually it's not a single layer. The external the punya layer is actually there is a jacket, and we call it water jacket. Okay, so you can see that actually this is the red tube here. This is where the water comes in, and there is also another actually water comes out. So that's how the temperature within the reactor can be controlled. Okay, so if you see the diagram here, so this is water in normally from the bottom and water up at the top. So this is how the temperature can be controlled, the mechanism of the temperature control of the reactor. And also it behaves as control the heat actually. It removes the heat. Yeah? It removes the excessive heat from the reaction, from the fermentation. Yeah? Okay, so that's water jacket. And this one is water jacket is one type of mechanism for the heat exchange and temperature control. Another mechanism used is by this one, cooling coil. Coil, this coil inside. This one is actually quite the same. Like if you see the boiler, the kettle, it has the coil inside, right? So that coil is used to provide the heat Tapi this one is not that really common in, I never see the reactor with this one, but there is a reactors with coil inside here, normally to, to control the temperature. The one that is common is water jacket like this. Okay? But it's just for you to know that there are different mechanisms for heat exchange and temperature control. Okay, and then the next one is pH control. Okay. Another important aspect of bioreactor is pH control. So how the pH inside the reactor is controlled. So this is pH probe that is inserted into the reactor. So it's through this top plate ni, dia ada lubang inlet for the pH probe. The whole thing here is called pH probe, right? So this pH probe is inserted into the vessel. So it touched the culture. So that's how it can read, it can give the reading of the pH. Okay. So this one is actually, if you autoclave it, this is the cap for it, the lid. And if it is after the autoclaving, we remove the cap and we connect the probe to the control unit so that we can read the pH. Yeah? So that's how it works. So when the pH probe, it reads the pH, it is also actually, the control unit is also connected to acid and base. So let's say we have the set point of the fermenter. Let's say we want to set the fermentation at pH 7, okay, pH 7. So whenever there is, so throughout the fermentation, there are the banyak byproducts produced. So that will affect the pH. So sometimes even though you start with pH 7, so if you don't control the pH might be reducing because of there are many organic acids produced during the fermentation. So it will reduce the pH. So how to make sure that the pH is always maintained at 7 at the set point. So whenever there is a reduction of the pH, dia akan baca, okay, dekat reactor, and then it will give an instruction for the, kalau dia reduce, dia akan bagi ni lah base, so that the pH increase back to 7. Let's say the pH kan, let's say from 7, it starts to reduce from 7 to 6.15. So bila dah 6.5 tu, the control unit tu dia akan bagi instruction for the base to be inserted into the culture. So dia ada, apart from the pH probe here, there is a acid bottle and base bottle that is also connected to the control unit and to the reactors. Okay. So the mechanism of the pH control is by the addition of the acid and base. So if the pH is too low, you want to increase it, you add the base. Okay? If the pH is too high, you want to reduce it, you add acid. Okay. Normally the pH, I think most of the cultures have a pH of 7. So that is the set point. Okay, so pH control is one of the important things in bioreactors. Other factors, the sterility, I talked about sterility just now. So you have to autoclave the reactors together with the media before the fermentation. After the fermentation is actually for killing any residual of the cultures. Temperature control, we talked about it. pH control, we have talked about it. Mixing as well, the agitation and the impeller just now. Okay, those are the minor factors. Maybe you can read by yourself. And sterilization, okay, the second last part, sterilization. Sterilization, okay, so as I said just now, sterilization is just a general process of destructing the microbes. So different methods can be autoclaving, radiation, filtration. So that are different methods. Okay, so for fermenter, how you sterilize the fermenter is by autoclaving normally. So this autoclave machine is you can see like it's like a washing machine, right? So this one is for small equipments. 2-liter reactor I think can be accommodated into this autoclave, but if you have big reactors, you have to use special types of autoclave, which is like a steam autoclave like this. Okay, so basically we use autoclave to sterilize fermenter and also the media inside the fermenter. Okay. Right. Another important thing about sterilization during the fermentation. Okay. So aeration, when we provide the air need to be sterilized because we know that air, it might contain particles that might not be, that can be impurities. Okay, so having said that, the air which is supplied to the reactor, this is just example of kalau the vessel tadi kan, the vessel, it must have this tubing with this sort of filters before the air can be provided into it. So meaning to say that the air will pass through this filter before it will pass to the cultures. Okay, so that is in terms of air sterilization. The purpose of filter is to sterilize the air from any unwanted impurities or particles from entering the cultures. Okay? So it's not just about just now the sterilization of the fermenter, the media, but also the air should be sterilized. So how to sterilize it is by filter. And filter ni, it has different pore size. We call it pore size is the one that on the membrane. So that will actually eliminate whatsoever the particles from entering. So if let's say you want to avoid bacteria, you have to know what is the size of the bacteria. Normally the pore size used is 0.2 micrometer. It depends on what type of particles that you want to avoid. So that pore size determines the particles that can go through. Okay. Normally for avoiding for this fermentation, we use 0.2 micrometer filter. Okay. Any questions so far? Right, medium sterilization. Okay, I try to speed up because it's already 2 hours. It's too much for you maybe. This is autoclave. Yeah. I think this is, I mentioned about the autoclaving just now. Yeah. So it's there are 2 methods of sterilization, batch and continuous. Batch is the one that you just put the things and take it when it's finished, right? Continuous is something that is continuous lah, something yang memang happen macam contohnya macam apa ni? This one is for sterilizing sorry, so yeah, is the medium is supplied to the sterilization chamber. So the sterilization process still happen. Sorry, macam ni. You provide the media, and then maybe you collect the media, but the sterilization process still happens. So that's why it's called continuous. Unlike the batch, you put the things in, and then when the sterilization process stops, and then you take it out. Okay, but that's the difference between batch and continuous. Continuous it means that the sterilization process still happening when you collect the your sterilized media or things that you put. Okay, the last part would be the forming. Okay, so forming is the formation of foam. So whenever you during the fermentation, even if let's say you shake something, like you can see the foam on the top of your drinks maybe. So the same thing, like during the fermentation, the vessel, when the things are stirred, so after some time there would be some foams created or formed on the top of the cultures. Okay, so what is the problem with these foams? If let's say the foam is not that much, it's not that really a problem. But if let's say the foam is very is extensive, it's quite a lot. So what happened is that, for example, like this one is your culture, right? This is headspace. If let's say the foam exceed this headspace, that will be a problem because it will flow out from, it will flow out. So it can cause contamination. Okay, so that is the undesirable thing lah. So how we want to control the forming inside the reactor. Okay, the first one is by having headspace. That's the purpose of headspace, is to allow some space so that if, let's say, forming happen, it will not that ni lah. Tapi kalau excessive forming, that one needs to be overcome with adding antiform. Okay, so one of the basic methods to reduce forming is to have the headspace. So the more headspace means the much better lah, actually. Okay. So I talked about headspace before, so I just take it quickly. Okay, and then the second way of eliminating the form is by adding antiform. So antiform is a type of chemicals where you add into the culture so that it will break the form. Okay, so that the form will not be exist on the cultures. So it's chemicals, animal and vegetable oils, silicones, glycols. So these chemicals can be used as antiform. So whenever you use this antiform needs to be sterilized as well. So not together with the culture, but it is in a separate bottle. So actually there is a tubing that will be connected to the vessel. So that's how the antiform is added to the cultures. The disadvantage of antiform, so there are some disadvantages. Reduction in oxygen solubility, so it might affect the oxygen transfer. So that's one of the disadvantage, but that's the thing that we have to compromise lah. Sometimes we just have to compromise with it, but as long as it doesn't affect that really much, it is still acceptable. Okay. Affects the separation and purification of the product. Yes, so because antiform is an external thing. Yeah, so apart from the thing that is produced inside the culture, when we add the antiform, is something like it's not impurities because it's sterilized, but it is a foreign thing, so it will affect the purification step at the end of the production process. Yeah? Right, another point is the microbe may consume antiform instead of the actual medium. So some antiforms, like oils, it can serve as food to the microbes. So it might affect the real substrate consumption during the fermentation. Okay? So that's one of the advantages of antiform, chemical antiform. Okay, another way how to eliminate the form is by using mechanical antiform. Okay, this one is by using external things lah, macam ni. This is Agitator deformer, so which is added on the shaft. You know the shaft, the long shaft that where the impellers are mounted. So this Agitator deformer, whenever the things are rotated, agitated, so that will help to reduce any sort of form on the top of the liquid. Okay, so that's mechanical method lah, mechanical antiform method. Okay. The disadvantage of mechanical antiform ni, it does not work. It does not work at low RPM. So meaning to say that it can only be used if the agitation speed is perhaps high enough, like maybe at certain more than 500 RPM maybe. So it might not be suitable for those cultures that need to be operated at low speed. Okay, so some of the microbes might be quite sensitive to shear, so we cannot apply high speed. But this one tadi tak boleh pula dekat low speed. Yeah? So that is the disadvantage, meaning to say that it can't be applied for cultures that needs to be operated at low speed, because it only works at high speed, this equipment. Okay, right. So those are the methods how to reduce form. Just now, by applying this rule of headspace, by having headspace, and then by chemicals antiform, and by using mechanical antiform, mechanical device actually. Okay, so that's it for this LU. So basically we talk about the design and features of bioreactors, right? What is bioreactor, fermenter? What are the important aspects of it? And then we have a look at the types of bioreactor: stirred tank bioreactor, air-leaf bioreactor, loop reactor, disposable, immobilized. And what are the fundamentals of bioreactors? What are the things that need to be taken into consideration, right? The design of the fermenters, agitation, aeration, heat exchange, mass transfer of the oxygen, pH control, right? And then how to sterilize the fermenter by using autoclave normally. The air supplied to the reactor needs to be sterilized using filter, right? The medium needs to be sterilized, need to be autoclaved, right? And another aspect is foaming, because during fermentation, foams will be formed on the top of the culture. So how to eliminate the foams? We can apply the headspace rule, right? So by having headspace, it can eliminate the overflow of the foam. And then by using chemical antifoam and mechanical antifoam. Mechanical antifoam is the device Ikato, so you put it on the shaft, but there are limitations for each of the methods. It depends on what kind of application that we are into. Okay, so that's it for today. Any questions before we disperse? Any question, guys?
Doctor,
Yes?
there is space at the chat box from Eden.
Oh, okay. Wait, hold on. Um, have to go into the chat box here. Wait, I'm trying to open the chat box.
The question is
Oh, yes?
to
Can
prevent
you read the question?
contamination. Yes. To prevent contamination, is it necessary to do inside laminar flow hood?
To prevent contamination, is it necessary to do under the laminar flow? Laminar flow, is it?
Do inside laminar flow hood.
To do what? What do you want to do? What do you want to do under the laminar flow? Okay. I cannot see the chat box yet. Sorry for that. Can you repeat the question again?
To prevent the contamination, is it necessary to inside laminar flow hood?
Okay, so for the bioreactor.
Yes.
Okay. So the bioreactor is outside, macam dia besar kan, so it's outside the laminar flow. So certain things, you just have to do it at the bioreactor itself. So what are the precautions that you need to make sure in order that you can avoid the contamination? So whenever you do the sampling, you have to make sure that you have the ethanol. You spray the things related to whatever you are dealing with, like the sampling pot. So to answer the question, yeah, we don't do the things under the laminar flow because the bioreactor is outside. It's big. Yeah, it's not the same as when you handle or you deal with shake flask. But the precautions that you can take is, as I mentioned just now, like you take the alcohol, spray whatever related things, and whenever you open it, you reduce the exposure. You try to do it like properly, quickly, especially for the sampling when you have to change the bottles and you have to take the culture tu kan. Another way is by having a flame lah. Sometimes we just, but I don't do it when it depends on the lab. Some of the lab, they just rely on the alcohol. That's what I did in my lab when I did my PhD, but when I did my master, I used flame in order to sterilize all the things that, for example, the sampling pot and so on. Does that answer your question?
Yes, thank you.
Okay. Okay, any more?
Doctor, I have a question.
Yes.
I read a statement that said that an aerated bioreactor will increase oxygen transfer rates with the addition of detergents because it discourages bubble coalescence. So does detergent count as antifoam too?
Detergents, antifoam. Yeah, normally, what kind of detergent?
It doesn't say it here, but it's just like biodegradable detergents, any types of chemical that will reduce the surface tension of the liquid.
Macam mana soalan ni? Sekejap. Antifoam tu? Can you repeat again the question? Because I don't get clearly.
The statement here said that an aerated bioreactor will increase
Uh-huh.
oxygen transfer rates with the addition of detergents because it discourages bubble coalescence.
Uh-huh.
So does the detergent count as an antifoam?
Detergents. Biasanya kalau detergent yang masuk dalam culture tu antifoam lah. Yes.
Oh, okay.
Right. Okay, any more before we disperse?